DNA repair mechanisms, I'll be talking about this in a little bit of detail. Before that, a little bit of basics. So we know that the DNA is made of a double strand DNA molecule. So we know that AGCT are the nitrogenous base pairs, where A and G are purines and C and T are pyrimidines, and A has to bind with thymidine, adenine has to bind with a thymidine with a double hydrogen bond, guanine has to bind with a cytosine with a triple hydrogen bond. These are our base pairs. Now base pair with a sugar, that is pentose sugar, is called nucleoside. With the phosphate, nucleoside with the phosphate is called nucleotide. Now this, if you zoom in, this is our nitrogen base pairs, and these are the bonds that it has been attached to, and this is the sugar, pentose sugar, and this is the phosphate. Together this whole is called the nucleotide, and this whole is called the nucleoside. So so coming to DNA repair as such, so DNA damage can happen, and damage has to be repaired. So the damage can be single strand damage or double strand. So both the if this is the DNA, either only the single strand has been damaged or both the strands have been damaged. Now single strand damage repair, what are the types of the mechanisms is base excision repair, nucleotide excision repair, mismatch repair, and homologous recombination repair, and non-homologous enjoining repair. Now little bit of detail about that. We know that A has to bind with T, G with C, that's how it has to go. So these are correct. Now you come here, A has bound with C, it has to bind with T. So now this will repair. So this is a mismatch, right? And also apart from this, there will be insertional or deletional errors that has happened during replication. So that will get repaired by MMR. Now apart from that, there is base excision repair, where due to oxygen radicals or hydrolysis, now normally we just need guanine. So if this guanine is has been acted upon by this oxygen radicals, the guanine will be you can see that 8 oxoguanine has been put here. So this has to be repaired. So this will be removed and repaired by this mechanism, that is base excision repair. Now due to UV lights, there will be ducts that are formed in the nucleotide, or pyrimidine dimers which has been formed. Now these are being repaired by nucleotide excision repair. Now all these three were single strand repair, DNA repair mechanisms. Now coming to double strand, here this is homologous. So it's like it has been damaged both the sides at one time. So this is homologous repair, recombination repair. So it has to just join homologously. So that is a homologous recombination repair. Now here there is it is non-homologous. So you can see this is very earlier, here it is delayed. So here it has to join it, these ends it has to join, but it is non-homologous. So it is non-homologous and joining repair. So these can happen due to ionizing radiation or anti-tumor drugs, chemo drugs, and everything. So these are double strand breakage repair, DNA repair mechanisms.